Hi, good afternoon. Thank you for the opportunity to share some of our experience of creating and using the animal models of CLL for mechanism studies and also preclinical studies. So chronic lymphocytic leukemia is a dynamic disease with many genetic and epigenetic changes in CLB cells. In addition, the microenvironment, including the exhausted T cells, supporting FRC and bone marrow stromal cells, as well as the myeloid cell that talk to B and T cells, all contribute to the disease progression. So there are two major types of animal models of CLL that people have been using to define the interaction of CLL cells and the microenvironment. One of those is the genetic engineered mouse model, and TCL1 transgenic mice is one of the most common one in this model. So this approach allows us to manipulate the genes that drive the disease progression. There is also the PDX model that allows us to transfer primary CLL patient cells into the immune deficient mice. So this will better reflect the complexity of CLL, as Nick mentioned earlier, but it also has is a lack of the proper microenvironment. So I will focus on TCL1 transgenic mice first. So TCL1 is an oncogene that overexpressed in the CLL patients. In 2002, Dr. Croce's group established these TCL1 transgenic mice. These mice have disease similar to human CLL, including the expansion of CD19, CD5 cells and the development of the lymph node and the spleens within 10 to 20 months. In this animal, there is a microenvironment similar to CLL patients, including the exhausted T cells and also the myeloid compartment that suppress the T cell proliferation and function. TCL1 mice also have AID expression only when the mice are sick. So this is one of the examples. This 14-month-old animal have a clonal expansion of CD19 and CD5 double positive cells. You can find the AID positive stem cells in the spleen. The AID expression in TCL1 mice is correlate with the cell proliferation, which is also consistent with the patient data that has shown the double stand Cas6-7 and the AID double positive cells in the patient lymph node. So Dr. Yan in our group have crossed the AID transgenic mice with TCL1 mice. These double transgenic mice have aggressive disease compared to TCL1. So these double transgenic mice have oncogene, both TCL1 and AID, specifically expressed in B cells, but not other cell lineage. In these animals, they have CLL cells enriched for VH11 and VH12 B cell receptors that binds to autoantigen phosphatidylcholine. Majority of these mice have this IgM clone, but you do find a switched IgG clone in some of these animals. And this is important, and this IgG clone has never been found in TCL1 mice. We found that the TCL1 AID CLL cells are transferable, like TCL1 mice. The adoptive transfer of these CLL cells into wild-type Black6 mice accelerated the disease progression, and that is correlated with the increased PTC binders. So these results suggest the antigen stimulation also promotes CLL progression in TCL1 AID double transgenic mice. Interestingly, in one of the transfers using the TCL1 AID mice, we found that the IgG clone can appear during the adoptive transfer. So this is one of the examples from the donor. So you can see the CD5 positive CLL cells from the donor cell. We injected those cells into Black6 mice, and you see the accelerated disease with the decreased CD5 expression. Using these cells, and then we perform another transfer into the Black6 mice. In 3 out of 5 mice, these mice developed IgG cloned tumor with decreased CD5 and the phenotype similar to the Richter-like cells that is similar to the patients. Interestingly, when we use these cells to transfer into the black 6 mice and treat them with ibrutinib, this line is resistant to ibrutinib treatment. So this is a summary of the first part of my talk. So T-cell1 disease progression is highly dependent on the tumor microenvironment. Overexpression of AID can be found in T-cell1. If you do the double transgenic AID and T-cell1, that can increase the mutations, which will be discussed later in another talk. And this will promote CLL disease progression and the transformation. Interesting, using the adoptive transfer system that I just showed you earlier, it provides a platform for mechanism and the preclinical studies. So one can use that model to study how the tumor microenvironment changes, especially the lymph nodes, in the BLAST6 wild-type mice, changes upon the infiltration of T-cell1 cell and the Richter cells. So we are starting that collaboration with Dr. Ellen Runstead right now. Also, final question is how you can find an effective treatment to prevent such transformation. With that, I will move on to the second model. So patient-derived xenograft PDX model, there are two major approaches as people have been mentioning. One is to directly use the PBMC to transfer into the NSG mice. The second one is to activate the T cells, as Nick mentioned, before transferring the autologous T cells together with PBMC into NSG mice. So we were interested in why the PBMC engraftment is relatively more diverse compared to the activated T plus PBMC in the NSG mice. We found that this is probably contributed by the different function of CLL fractions. So this is one of the examples of the flow. So if you check the fractions in the CLL PBMC taken from the blood sample, you can find the cells that have the highest Ki67, which is called the proliferative fraction. They express less IL-10, less higher IL-4 producing B cells. In addition, they produce CCL3 and CCL4 cytokines that can recruit T cells in the spleen and then further activate the T cells. So this is the animal that injected with only the purified proliferative fraction together with the autologous T cell without pre-activation. So you can see these PF cells can activate the T cells and then further support the B cell engraftment. In contrast, if you inject the mice with only the resting fraction and the intracellular fraction, which is the bulk of tumor, these two fractions express higher IL-4 producing B cells and less IL-4 B cells. They tend to have no B and T cell engraftment in the animal. If you mix the fractions, if you mix the PF with the IF or RF, you can find the robust engraftment of PF-mediated CD20 and CD3 cell in the mouse spleens are absent. This suggests the fractions have different function for the B and T cell interaction. So the activated T cells are more robust, give you more robust B and T cell engraftment in the NSG mice. Right now, this is a standard approach we are using in the lab. So you pre-activate the T cells from the CLL patient after selection. After 7 to 10 days expansion, you added autologous CLL PBMC, and we are now more oftenly use the IP injection as an approach. You can pre-treat the NSG or SGM3 mice with busulfan before the cell injection, and then euthanize the mice depending on the purpose of your experiments. So this is more consistent engraftment of both B and T cells using this approach. So using this approach, we have done several preclinical trials. This is one of the collaboration with UPenn. So here I'm showing you 3 patients that we send the PBMC samples to UPenn to generate CD19 CAR T cells. And then when we received the CAR T cells back, we took the PBMC cryo vials, we selected the T cells, and we did the same thing, we injected into the animal. And 10 days after cell injection, we injected the mice with 1 million CAR T cells by IV, and then we just leave the mice there for 3 weeks. So what happens is in the control mice, you have, compared to the control mice, the treated TDN is the CAR T treated mice. You have more T cells. This is mainly the expansion of the CD19 CAR T cells, that's CD8 cells. And you can see consistently decrease of CL cells in the spleen. This suggests the animal model is good for the preclinical trial, and this is another example. So here we use I'm showing you one case of the patient that was treated with ibrutinib for total like 7 years. We took the sample when the patient responded to ibrutinib, and we also took the sample at the time patient developed the resistance to ibrutinib. And then we treat the mice with either vehicle or ibrutinib. We found that in the ibrutinib-sensitive cell-injected mice, the ibrutinib treatment is able to suppress the CL B-cell engraftment in the spleen. In contrast, the ibrutinib-resistant mice, first, they have less T-cell engraftment percentage-wise compared to the ibrutinib-sensitive cell-injected mice. Secondly, these cells are resistant to the ibrutinib treatment. So this again suggests that this animal model is good for preclinical studies and maybe mechanism study. This is the final experiment I'm showing you. This PDX model is also good for Richter transformation studies. This is one patient before and after Richter transformation. So you can see the decreased CD5 as I showed you before in the T-cell and AID mice. Comparing the Richter to CL, we found that the Richter cells have higher CD52 and the CD20 expression. So we have this collaboration. Nick has this collaboration with Scripps that they established the CD20, CD52 bispecific antibody. So this experiment was taking the Richter patient PBMC, select the T cells from Richter PBMC, and then inject the mice after activating the T cells together with the PBMC. 10 days after, we did the bispecific antibody IP injection. 3 weeks after, we sacrificed the mice. So again, the Richter cells in the SG mice have reduced need for T cell support. You can see the T cells are not expanding as much as the CL cells here. With the drug treatment, you can find the CD20, CD52 bispecific antibody. Here showing you 3 animals that have very efficient eliminated the CLB cells. So for this part of my talk, I show you the PDX mouse model that allow us to study the crosstalk of CLB cells and T cells that suggests the immunosuppressive microenvironment and the immunostimulatory cell proliferative fraction. You can use this mouse model for preclinical studies and also to transfer ibrutinib-resistant cells to study like drug resistance and sensitivity. And also this model allow you to engraft the Richter patient cells that have very complex genetic changes. So with that, I'm moving to the next last slide. So there are, with all these good things and bad things that we have seen in the models, there are many other directions we should consider. So for the genetically modeled modified animals, we probably want to consider developing a mouse that can reflect specific human CL subsets, and a model that mimics the microenvironment features. For the xenograft mouse model, we probably want to think about the approaches to identify the genetic basis development and the involved evolution of the CL. So with that, we can probably do different source of the cells like stem cell, MBL cells, or inject the animal with cells originated from spleen or lymph nodes. And we should consider how to improve the microenvironment, especially the myeloid compartment, like Nick mentioned. And probably we can have the recipients with more defined microenvironment. And the different route of administration might be one of the other ways that we should consider. So with that, I want to thank people in the Feinstein Institute and our collaborators. And thank you for your attention.
Thank you, Shih. This paper's open for discussion. Anybody got any questions?
Maybe I can start with one.
Yep.
So you mentioned you can use the xenograft models to study clonal evolution, where you just mentioned evolution. But how can one envision that you study clonal evolution? Because the time frame in Nick's talk was about 124 days only.
So it varies actually. The slide that I show has the mouse that have this aggressive clone that kill the mice in 21 days. But there are clones that can, there are clones in the animal after adoptive transfer can have the mice survive for 60 days. So depending on how the clonal evolution occur in the animal and what the donor cell has the different clones, the mice actually have different survival after adoptive transfer, between 20 to 60 days so far, that's what we saw.
Anybody else got a question? So Shih, just one from me. So in your double transgenic mouse, the AID TCL1 mouse, do you see the sort of classic AIDS-driven mutational landscape in those mice?
So that is actually the talk that we will, the AID talk that they will explain better. Yes, there is the increase of target mutations in those animals,
Fantastic.
You will see those data later.
Thank you, Shih.
Thank you.